We're going to continue with computed tomography. And then here's an example of why computed tomography, for example, has taken off over the last 30 years and is now done in sort of 100 million people a year. And so here's somebody who shows up in the ER, has acute chest pain, but is a young person. So it's highly unlikely they have a coronary event. They're not having a heart attack. They're like 32 years old, right? But they've got acute chest pain. So what do? Put them in the CT scanner, inject a contrast dye to look at their chest. And so we get this composite view where we have bones plus soft tissue, and the really bright stuff down here is vascular tissue, or the vessels themselves, right? And those with experience would look down at the aorta here and see this line, the very dark line that's going down the middle of the aorta. And on the next view, you take out the bones and you look in her aorta from the back, and there's this dark line all the way down. This is called an aortic dissection. And what has happened is the wall, the epithelial cells and the wall of the large vessel have peeled off of the wall and are now sort of going out halfway into the aorta. So there's one side of this structure that still has blood flowing through it, and there's one side that's just kind of a bubble inside there. And so this changes your life instantly, right? You need emergency surgery right now because your probability of dying just shot up when you are walking around with this thing in your chest, right? So this is the reason why when people have car wrecks or they fall off a ladder or they fall down and hit their head on a ski hill, first thing you should do is go to the hospital, get a CT scan, right? Because it tells you what the heck's going on. And and this scan, remarkably, is done on a modern, this happens to be a Siemens scanner, probably, at under 3 millisieverts of radiation. You don't really know what a millisievert is, but walking around San Diego for 1 year and living in your house with radon, with radiation, like you get 3 millisieverts a year just being alive, right? Just sort of walking around, right? And so this is the, sort of background radiation level to produce these pictures, right? And the pictures themselves were produced in 700 milliseconds, right? That whole thing, right? So this patient was flying through, the spinning detector at that speed to get all of this data in under a second, right? It's absolutely spectacular technology, right? And God only knows how many people's lives it saved, like tens and tens of millions of people, right? So accordingly, say from 1995 through 2007, this is the number of hospital visits that included a CT scan, okay? And this is the percentage of those visits that included a CT scan. So you can see from 95, where you had about 2 million, through 2007, we're up to 16 million. This is probably just Medicare data because the numbers are fairly small, but the growth curve is the same across all, providers. And now we're up to about 90 million CT scans a year done in the United States. Just let that sink in for a second. How many people are in the United States? There's 340 million or something. 90 million CTs are done, right? That's a huge, significant chunk of the population is getting into a CT scanner and getting scanned, right? So it turns out, obviously, that CT scanning becomes a relatively large component of background radiation that everybody is exposed to. And the question is, does that increase your chances of getting a cancer? Right? Because a X-ray that has an energy of, say, 100 keV or 140 keV, and we'll look at that, has the opportunity when it goes into your body to cause chemical changes. And one of those chemical changes is it can cause a double-strand break in DNA. That is usually repaired right away, right? 99.99% of the time, it's just going to get repaired, right? But the repair can screw up, and then you have a a somatic mutation in your genome in a very local spot, right? So it's not your germline hasn't changed, but the cells that underwent this transformation have changed their genetic makeup. And it's pretty clear that some of those genetic changes really promote cancer. Right? So here in 2018, 90 million were performed. The increase is pretty uniform. And then by exam type, you can look at this data later. These will all be posted. You know, it shows you sort of the ratio of where those scans are done. This is a CT image of a heart. And so everything's been taken away except the primary heart itself. And these are the coronary vessels in this patient's heart. And so you have two primary coronary vessels coming off your aorta. The LAD, which is this one's left anterior descending coronary artery. This is the circumflex, goes back around here. This is the right coronary artery. And this cause, abnormalities in these vessels that occur over time. As you get older, you develop basically lesions on the inside of these vessels, right? And they can get fibrotic, they can grow little bits of calcium in them. And so eventually your vessels get smaller or shrink, they get narrower and narrower until you can have a crisis in which the vessel is so small that blood does not really perfuse the tissue downstream, and that's a heart attack. And that's the primary cause of death for most people, right? When you get old enough, eventually either you're going to die of a heart attack or cancer, kind of. That's like 70% of people die of one of those, 50-50 split, maybe a bit more. My wife does cancer research with genetics. And I'm always like, well, if we can eliminate cardiovascular disease such that you don't have a heart attack till you're 100, then everyone will die of cancer. Which, until we eliminate cancer, which is another thing you've got to eliminate. Okay. But if you look at the resolution of this thing, it's pretty spectacular, right? This picture, the whole heart was imaged in 150 milliseconds, right? To do an MRI image with this kind of resolution, each one of the voxels, they're like little bricks, 3D bricks, and they're 1 by 1 by 1 millimeters is the kind of basic spatial resolution of this, but it's displayed with more bricks than that. But if I was to do an MR and get that same voxel size, it would take 4 hours, right? To get half the signal to noise I'm seeing here. It's just, there's so much less signal in MRI, right? So I refreshed this. This is a new slide, slightly new slide deck. So I'm going to refresh the one that's on the website. So these links now are better and work better. The way to appreciate just how good CT is go to the marketing, a branch of the Siemens, GE, Philips, Canon websites, and look when they say our image gallery, just look at the spectacular pictures that are there. Okay, MRI, basically the physical phenomenon of nuclear magnetic resonance was discovered in the mid 1940s. It was discovered simultaneously on the West Coast and East Coast, and these guys shared the Nobel Prize for discovering it. And it basically is the following, that if you have a proton or any spin 1/2 particle or even an odd spin nucleus, but a proton has a spin 1/2 nucleus, when you put it in a magnetic field, it turns out that the orientation, it can have one of two orientations in that magnetic field, the actual nuclear spin, right? And electrons also have a 1/2 spin. And if you shoot electrons through a magnetic field that has a gradient, they will make two beams, one spin up, one spin down. So they can only be in one of those two conditions. It's pure quantum mechanics, right? And so what happens is the nucleon, like the proton, say it's in water, and you put the sample in a magnetic field, you get almost half of the protons are in spin up and half are in spin down. There's a slight increase in the number that are in spin down because it's a lower energy state, and the thermodynamics of the situation just set it there at its equilibrium. So you have this slight excess of, spin down. And it turns out that the energy to convert a spin down proton into a spin up, like to flip the spin at, say, 1.5 Tesla, which is the magnetic field strength of an MRI scanner, that energy is equivalent to a photon that has a 64 megahertz frequency. So it's a radio wave, right? And so with these radio waves, when you put it in at exactly that frequency, such that that energy is achieved, the transition between spin up and spin down, you see all of a sudden you'll be irradiating a sample, say with radio frequency energy, and you have a frequency dial, and you start at, say, 55 megahertz, and you turn up the frequency dial slowly, and you watch how much RF is being absorbed by your sample that's sitting in this magnetic field. We turn it, turn it, turn it, turn it, and then at 64 megahertz, right, to like 6 digits in the frequency, all of a sudden, whoom, all of the RF starts going into the sample. It gets absorbed selectively by the sample. And that's called resonance, right? And that's this nuclear magnetic resonance phenomenon. And that's how they used to do it. They used to sweep the frequency of the RF across and look for this dip, such that the sample itself, all these spins start changing their state because they're in tune with the RF. And so it was Lauterbur, a few people were responsible for taking this phenomenon and making it into imaging. It's not something that you would automatically think you can do imaging with, because the radiation, it turns out that if you irradiate your sample and then you hit resonance and you irradiate for a short period and then stop irradiating the sample, but you keep the antenna there and you listen, then RF comes back out of the sample at that frequency, right? And so that's the signal you're measuring. It's an RF signal at 64 megahertz at 1.5 Tesla for water. And so, we've got this, the patient is actually glowing in RF. They're irradiating this. So that's our signal. And then you ask the question, can I make a picture with that signal? Maybe. But what's the wavelength of a 64 megahertz photon, right? Anybody have a guess? It's 4.5 meters, right? So you got a wavelength that's 4 meters. So how are you going to image with that thing, right? Like in optics, you need shorter and shorter wavelength photons to get higher and higher resolution of the object, like to reflect off things, right? So we got to do some other kind of smart imaging here, obviously, than that, right? So this guy figured it out, one way to do it. And it was basically, he has his spectrometer, and when you do these nuclear magnetic resonance experiments, you look at a lot of different frequencies coming out of the sample because there are many different nuclei in there, et cetera. And to get it to work right, you really need to make the magnetic field super uniform across the whole sample. And so in order to do that, the manufacturer of this thing gives you a primary magnetic field and then a whole set of gradients. And there are coils that change the magnetic field slightly, spatially. So it'll make it higher at the top, lower at the bottom. And so you go through about 12 different adjustments to try and make the magnetic field as absolutely uniform as possible over the whole sample. And that way you get a more coherent signal, right? Those are called magnetic field gradients, and they showed up in NMR spectroscopy in chemistry. This guy was a chemist, and my hypothesis is he was really terrible at adjusting those dials, right? So think about it. It's a 12-dimensional optimization. I've got to adjust the current in each gradient coil to get my magnetic field as uniform as possible. Some of them are linear in X, linear in Y, linear in Z, and then you have a quadratic term in X. You can have a third order term. And so you've got this whole bank of dials. That's not an easy optimization problem, right? Just like, what setting do I put each one of these 12 dials to get my best thing? That's a hard problem. I think he was terrible at it, and he was just like, I'm just keep going on this dial and see what's going. So he just keeps rolling up the X gradient, and then he looks at his spectrum, which should have a single frequency for all the stuff that's in his test tube, right? Should just be a spike at whatever the resonance frequency is, the stuff in that test tube. But he's cranked his X gradient to such a terrible value that across the sample, I have low frequencies on one side of the sample and high frequencies on the other. And he looks at his spectrum, and by God, it's a profile of the test tube, right? It's like, it's a thing that looks like this. Like one side's got these frequencies, one side's got these frequencies. So he's projected the spatial dimensions of that test tube onto these frequencies purely by accident. It would be nice to be able to ask him, but he's not alive anymore. So I'm sticking to this story. And then, so he does that with two samples in his spectrometer and makes an image of here's one sample over here, one sample over here. I separated. It's not just one frequency coming out with the gradients. I can tell you where those things are. Right? It's really, really cool. Right? So here's some of the early adopters of this. Here's a guy. His name's Raymond Damadian, and he built one of the first human-sized scanners. He made a magnetic field, big old resistor field, and then this RF coil around his lab assistant there. And his idea was he was going to measure relaxation times. We'll understand what those are by the end of the course in cancer versus non-cancer, and we'd be able to tell whether people had cancer, right? That was his big deal. And then here's this guy's Bill Edelstein. He worked at GE a long time ago in the 80s, but this is when he was a grad student or a postdoc, I can't remember, in Aberdeen, and they built a human-sized magnet and put one of the grad students in there with an RF coil around the grad student and gradient coils. So it's like making a human-sized NMR spectrometer, right? And so a lot of the original ideas for NMR imaging came out of these labs in England mostly, or Scotland. Excuse me. This is Peter Mansfield. He won the Nobel Prize with Paul Lauterbur. Those two got the Nobel Prize. He did not get the Nobel Prize, and he was really upset about that. But he was also kind of crazy. And I think if you're a nice person, you have a much higher probability of the Nobel Prize than if you're a real jackass, right? And he was kind of a jackass. So I watched him give a talk when I was early, like first year grad school or something, and he came up and gave a talk and just blistered the other people in the field as stealing his ideas. And he was just like, what is this? Is this what science is? It was like, yeah, he was crazy, crazy dude. Okay, so a modern MR scanner, this is a really big one. This is probably a 7T MR scanner, but the patient lies in a large static magnetic field, okay? And the field is, 50,000 times the Earth's field. It's a large field, okay? And normally that field is provided by superconducting coils that you put them in liquid helium such that the material itself has no resistance. You ramp the current up slowly such that it's stable, and then you click, shut that loop. You're in here with voltage, you go click, and you shut it, and then it's on all the time, right? So you're sitting there with a 7 Tesla magnetic field, and it never goes off, right? If it does go off, it's a bit of a crisis. So, and then on the inside, we have an RF coil that will both transmit RF into the patient and receive RF back from the patient. And then there's a set of coils just outside that, which are called gradient coils, and they're going to change the magnetic field as a function of position, and they'll do it in a temporal way. You can program how to change them as a function of position. Over the course of, say, your 50 millisecond acquisition, you can switch these fields around. So that's the prototype NMR scanner. And here's a typical 3T MR scanner. This is a picture of one that Tom has, I think, just over at at his center. And then these are crazy scanners. This one was at the NIH. It's a 12 Tesla, and this one was built in France, another 12 Tesla. And so the question is, why build them bigger and bigger and bigger? This seems crazy. It's a very American approach to this. Like just big a builder, so or build it bigger. Let's see what happens. So the reason is you get more signal, right? It just goes up. It's like with field strength, your signal to noise goes up. You also get some extra effects that are dependent on sort of spatial gradients in the sample. And in particular, for doing functional MRI, where you're watching the local magnetic field change in the brain, and it changes the relaxation times of the brain tissue and the tissue around small vessels. That's how you do functional magnetic resonance imaging, which is like fMRI, where you get somebody to do a cognitive task and watch their brain change. It's pretty cool, right? Um, so if you have time to get the data, and we're talking tens of seconds to tens of minutes, you can make absolutely spectacular pictures of soft tissue with MRI. It is absolutely beautiful. And so the image here is proportional to a constant we'll learn about called T1. And then you just change your imaging parameters. You have about 20 different imaging parameters you can change. You can produce another image from which the signal of different parts of the brain are different, but it just produces different signal, right? And so you get a new image of the brain. It's almost like this orthogonal image or in a different space. This image is proportional to the proton density. So we see the dark stuff here, there's fewer protons. It's less like water than, say, the cerebral spinal fluid in there has high proton density, right? And then there's a time constant called T2, which we'll hear about a lot. And T2 is really long in the cerebral spinal fluid. It's shorter in, that's my mother-in-law. Yeah, not going to take that right now. So those look like orthogonal data, right? If you look at the the CSF and T1 versus T2 and things like that. So you have a multi-dimensional ability at each voxel, right? It puts you in a space that has a higher dimension than just grayscale, right? This here's some images at one of those crazy 11.7T scanners. And, a whole bunch of us, I was in MRI for like 30 years or something. And as they kept on going up and the price kept on going up for these, like, what are you going to see at 11T that we haven't seen at 7, for crying out loud? And then you make the picture and you go, oh, okay, I see. Okay, that was worth it. Yeah. You see these extraordinary little details that you just did not see at 3T, especially. Yeah. Oh, okay. So the question is, when I say each voxel has a higher dimension than just grayscale, what does that mean? And so that means if I take a point here in the brain and say, how do I characterize the tissue at that point? Instead of just one number, right, which is the signal intensity of this image at this point, I now have 3 numbers that I can characterize that tissue point. I have whatever the T1 weighted picture is, I have the T2 weighted picture, and I have I'm sorry, proton density weighted picture and the T2 picture. And so if I plot, where that is, it's a 3-dimensional space, right? So it's a point in that space. And so now if I wanted to segment a certain type of tissue. Let's say I wanted to segment gray matter from white matter in the brain. So a gray matter tissue, as you can see, this is the best for gray-white determination. So this is all basically white matter tracks in the brain, the dark stuff there. And the gray matter is this brighter stuff out here on the cortex. And so if I wanted to make that classification, I don't have to just do it on a one-dimensional axis and the brightness on that axis. I do it as a cluster of points in space. Correct. So the question is, could you think about it as RGB? Absolutely. So each thing would have its unique color once you mix the R, the G, and the B, right? Or you could do whatever, you know, orthogonal you want. Okay. Now, MR has many dimensions. The other thing we can look at in the voxel is the diffusion constant of the water in that voxel. We can look at the flow of protons through that voxel over time. Is it in a space that's getting perfused heavily, or is it not? Many dimensions turn up. So here's an MRI scan taken of the vascular structure in a human brain without contrast agent. So we didn't inject a contrast agent to do this picture, right? Whereas it looks like an X-ray angiogram in a way that we inject a contrast agent. Here, what's done is you take thin slices and basically where there is a vessel, blood will flow into the slice and make it brighter because it's outside where you did your original excitation. And so this is a type of angiography. There's many different types of MR angiography. Here's perfusion imaging. So here, these, look like proton density weighted scans or T1 weighted scans. In the same position in that person's brain, I get this scan, which is blurrier, right? However, what the brightness represents here is the perfusion, the blood perfusion of that brain tissue. And that can be, so the brightness here is proportional to basically milliliters per cc per minute, right? So it's how much blood washes through that volume per minute. And you'll make it brighter if there's a lot of blood washing through there and less if there's less blood, okay? So if you dynamically want to see how is perfusion going in this person's brain, if there was a really dark spot there that was not symmetric, remember we're all nice symmetric down the middle, then that would indicate that there's a perfusion deficit. That person is possibly having a stroke at that location, right? MR can be done if the patient can hold their breath, and this patient has probably held their breath for about 12 heartbeats. I'm sorry, this is all not running smoothly, but you can see you can take MR data over the course of the R to R interval of the heart. And so what we'll do is we'll take the EKG while we're imaging, and we'll piece together data from the beginning, the mid, and the end of the ECG interval and just make a movie of that. So usually you make about 10 to 20 time frames, and you can just see what the heart looks like over time. Very useful if you want to figure out whether or not that person's heart is actually ejecting the amount of blood it should be, what the actual muscle, is there any of this muscle that's not working well? You can then just change your orientation of the scan plane interactively on the MR scanner. We have these 3 gradient coils, X, Y, and Z, so we just change current in them, and the imaging plane can just, basically it has those degrees of freedom to move around. So here we're looking at the myocardium of ejecting blood out of the left ventricle. This is the left atrium, the right ventricle. This is fat. Over here, the bright stuff. There's the spinal cord there, the descending aorta. This is fat. All of this tissue here, that's muscle, right? So it's pretty cool. And the interesting thing is, I don't know if it's projecting well, but you can see that there's kind of a circulation kind of signal there, the blood's sort of rolling around. And that tells you that circulation is reproducible from beat to beat, because to get this image, you had to average 12 beats together, right? So if it wasn't reproducible beat to beat, it would just show up as a mess, right? And then here's this functional MRI thing where you focus your attention on a small region of the brain, or you image the whole slice. And then as you're imaging that slice in time, so this white curve here is the intensity at a specific pixel in this person's brain. And then the green and the red are a stimulus being turned on and a stimulus being turned off, right? Or different polarity of stimulus. And you can see when the red stimulus is on, the signal on that brain seems to go up, right? And it doesn't look like much, right? It's a small percentage change, right? However, when you do it over time and you correlate these things together, you can get very good information about brain activity. And then the diffusion of water inside the brain can be measured, and the diffusion will be faster along the directions of neurons than in the orthogonal directions of neurons. So you can think of a bundle of neurons as kind of, say, a set of hoses put together, and they go down like this, right? So we're going to diffuse quicker in one direction than the other direction. So you can use that anisotropy of that diffusion. You just make a tensor, look at the direction where it's diffusing fastest, and you track that direction, and you build these maps, which are essentially neural connection maps. These are just the pathways of the neurons in the brain, right? So it's remarkable. So, okay. Any questions about that? So last year we had the class asked tons of questions, and it actually was good, right? I think you can watch these lectures in 2x or 1.5x, from last year or this year, whatever. But I think the questions really helped, like for us too, like what is it that isn't understood and what's most interesting and stuff like that. So please don't be shy. Just put your hand up and ask questions, and we'll answer. So here's 3 different imaging modalities. On the left here is a CT image, and the contrast window has been set such that the bone is highly visible and the soft tissue all looks the same, essentially. This is MRI, and it's probably a T1 image of the brain, not of the same brain or the same slice, just different looking picture of a human brain. And then this is a PET scan of a human brain. So this is the activity of essentially a proton emission tomography where well, we won't go into it right now, but it creates radiation coming out of the patient at 511 keV photons come out of the patient, you image them. Now you can see they're quite different, right? Contrast is different. Spatial resolution is different. This one's a lot blurrier than this one. And so how do we characterize these pictures? And what are the quantitative things we're going to say about the pictures? So, well, these are the fundamental things that we need to be able to discuss. Yeah? Oh, so the question is, can the PET picture give you any information that CT and MR cannot? And the short answer is absolutely. And what's done is the PET agent that creates these photons coming out of the patient is usually combined with some kind of biochemically active molecule that gets taken up by cells in a preferential way if those cells are neoplastic or those cells are, some other type of cell. So for looking for tiny lesions, specifically cancer. So if you have a primary lesion, say in your brain, and they want to know, is there any metastasis anywhere else in your body? You can make one of these PET images, and you'll see tiny little metastases will show up as bright because it accumulates that substance, right? So that's principally what's done. It can measure blood flow to different brain parts absolutely accurately. So if you're concerned about brain perfusion for people who have had stroke or something like that, it's also very good. 280C, we look at a much broader swath of imaging modalities, and we spend a couple lectures on PET and nuclear medicine. So contrast is just what is the relative brightness of different pixels in my picture? Like Some are bright, some are dark. And so we're going to quantify that brightness and darkness with contrast. Spatial resolution is the focus. Like, can I focus this thing so I get sharper edges? And so we'll look at definitions of that. Temporal resolution, does it take 10 minutes to make this thing? Does it take 10 milliseconds? That's kind of the range we're looking at here. And what happens if it takes me 10 minutes to make it in terms of breathing motion or cardiac motion or just the long-term stability of the patient themselves, like keeping in the same position. Noise is reserved usually for a random signal that's on top of your picture. Okay, so it's if we'll take a look at some examples, and signal to noise ratio we'll look at is just how bright is my signal of my target. If I'm looking for something, how bright is that signal divided by the noise level, right? We'll see how to calculate that. And then contrast to noise is if I have two signals, a background and my target, what is the signal difference between those two divided by the noise? Sometimes the noise is different on each one of those signals. The bright thing can have higher noise than the dark thing, etc. So we have to look at that. So this is straight out of the textbook. So the whole concept of modulating the brightness in space to make a picture, we're going to go back about an hour where we were talking about Fourier and talk about the brightness as you would model it with a sine wave. So here we have probably the simplest picture you can imagine, which is every pixel in this picture has the same value. Okay, so it's a uniform background. There's no features in that picture at all. And then looking at this formula down here, I'm going to modulate my original background, which is just A. I will then add to it this function, which is changing its amplitude as a function of space, right? So if I look, my spatial variable in here is x, and then I have this 2 pi u naught that tells me how does the signal oscillate as I change x, as I move in space, right? So it's called the spatial frequency, this constant right here. And so now if I change my values of a and b such that I now have 0.2 as b over a, right? So b is no longer 0. Then I see that there's a small modulation with that spatial frequency. And that's our new picture, which has a very simple structure. It's a plane wave. It's a sinusoid going from left to right, and it has a certain amplitude compared to the baseline, right? So our baseline now is hit only at these transition zones here. We go to peaks and valleys. And then I crank up my modulation or contrast such that now A over B or B over A is 50%. And that modulation now is 50% of the background level. And then here it's 100% of the background level. So this is basically it's going all the way down to 0, all the way up to 1, 0, 1, right? Simple. It's a plane wave. You can characterize this picture with just the amplitude of that plane wave. Now remember, from Fourier theory, we can build any function we want in this space by changing the frequency of sinusoids and adding them up together, right? So this is just one of them, but we can make any picture we want by adding these different sinusoids together. And not only in the x direction, but in the y direction as well, and at different angles. We can put this plane wave at different angles. So in looking at what our imaging system is able to achieve in terms of imaging an object, as you can imagine, any imaging system, if I put a test pattern in front of it, let's say that test pattern is parallel bars, okay? and I bring up my imaging system, I take a picture of the parallel bars, then when I look at my picture, the outcome, I see oscillating signal with the bars. Now what I'm going to do is I'm going to move those bars closer together, cut them in half, move them closer together. Take a picture again. This is just like the example we did before. And I look and see, what does my picture look like? It also has contrast between those bars. Eventually, as I make these things smaller and smaller and smaller and smaller, eventually, when I take my picture, I don't see any modulation anymore because they're too close together. I can no longer resolve them. And that's always going to happen, right? Because you always can make a sharper and sharper object that your imaging system can't manage, right? And so the transition from going from a simple sinusoid that we can image easily to something we can't image at all is essentially how we characterize an imaging system. How does it make that transition to where I can't image at all anymore, such that the frequency is too high? So mathematically, to look at that, we say, I have an input function here, like my input is the underlying object itself, right? And then I take a picture of it with my imaging system, whether it's my camera or my MR or my CT scanner. And what comes out as the picture? And as we saw, we modulate the amplitude, right? As it processes through my imaging system, the amplitude gets changed. That turns out to be different for different frequencies. As the frequency gets higher, this output eventually shrinks down to zero, right? So let's look at some quick examples. Here is this is a phantom that we had built in the machine shop here, where the machinist was fantastic. Let me show you the better copy. He drilled these very small holes of a certain diameter, and then one diameter gap, and then a new hole, and just put them closer and closer together as we go down here, right? Until finally these things are about 0.3 millimeter diameter and that far apart, which is very hard to do in plastic. To drill that without breaking your drill bit, I think it went through many drill bits, right? Anyway, so let's look at, here's a picture, and we say, oh, look, I can see an oscillating pattern in this direction. So I'm resolving the fact that there are, 5 holes there. I can see that. When I go down here, I say, well, I don't know if I can resolve how many holes are there, or if there are holes at all, right? And that's because the frequency, the spatial frequency of that pattern is now higher, and it's beyond our ability to resolve, right? If I go back to this scan, that lack of resolution happens much earlier. It's up here, right? And so whatever we're doing with this scan makes a much worse picture in terms of spatial resolution than what we're doing with this scan, right? And what we're doing with this one is we're imaging with very few photons. So we'll learn what this means. This is 96 milliamps of current is going into the X-ray tube, right? And it produces this picture. Now I crank the current dial, and now 380 milliamps of current is going to my X-ray tube. I make a lot more X-rays, right? And so when I detect those X-rays, I get a much higher signal to noise on my detector, and I'm able to resolve these things. The reason that happens, it's a little subtle actually, is when you're back here with few photons, it turns out you need to average those photons together across detectors in order to get a sensible signal at all. Because if you don't measure a photon at all in one of those detectors, that's a problem. Because then you have no idea, right, how many get absorbed. You just have 0 got through, right? So you can't really have 0. You have to do something more sensible than that. And then normally what you do is you spatially average things on the detector, and it produces a lower resolution picture, right? You can quantify the modulation here. So this is like the contrast modulation of this signal at a specific spacing, right? So we look at the peaks and the valleys and we see what is that modulation. That tells you how well that spatial frequency is getting through the system relative to other frequencies, right? And we'll do this as an exercise in the problem set. If I look at the same feature, the underlying feature is the same thing. I know exactly what it is because we drilled it. And then here's the picture of that feature. So now that modulation is kind of gone. You don't really detect the pattern at all. So yes. Okay, so the question is, how do you determine how many pixels you get in your final image, right? And you want more pixels than you have underlying spatial resolution. So we'll actually answer this more precisely in a few slides. But principally, it's like, how many pixels does my detector have? You know, if for CT, if I have 1,000 across here, right? How many pixels do I need to actually uniquely map to that picture? And it's usually, for CT, we're usually between 512 by 512 to 1,000 by 1,000, somewhere in there, right? But you want enough pixels to be able to resolve the point spread function of the image. And we'll get to what that means, okay? So another way of looking at resolution, and this is another simple test you can do with your system, is ask the question, what does the signal that my imaging system produces look like for an object that's really tiny compared to the detector? Okay. So now if I have a CT detector, and you know they're usually elements that are about 1 millimeter wide, and they're all stacked together, right? And so I'm taking projections, and it lands on that detector, right? So if I put an object in the scanner that's really tiny, such that it only goes in one detector, right? It doesn't spread out. What does the picture look like, right? When I image that, and so that's an image of a point. Practically, the way we image points in CT is we take very good Japanese pens. They're like 0.38 millimeter Pentel pens, right? And you sacrifice the pen. I'm sorry, it's a terrible thing to do because they're beautiful pens. But you bust the top off, and the little tungsten ball bearing comes out, and you can put that on a piece of tape and then stick it in the scanner. And it's a very small 3D object, right? And so like, here's one here, this guy. And so here, I like this definition of resolution. It's called the Raleigh definition. When I make my picture with my scanner, I don't see one pixel that's bright, or I shouldn't see one pixel that's bright, right? I should reconstruct the picture with a pixel size such that when I image something like that, I see a shape that is the point spread function, right, of that image. And so I make my pixels small enough so I see that shape, right? And it usually, ideally, that shape looks like a normal distribution or a Gaussian, right? So it's just a Gaussian function. Gaussians are great because their Fourier transform is a Gaussian, which works really well. They're well behaved in terms of a peak and smoothness, etc. And so here we go. I've got this thing in the scanner. It's this big, but the picture I get is this big, and I could have a lot of pixels along here showing that shape. And what we'll do is we'll take those two dots and we'll bring them closer and closer together, right? And ask the question, when can we no longer say it's two dots, right? So as I bring them together, these two Gaussians come together, and they add up to give me one signal, and it looks something like that. And you say, well, is there two dots there now? And so that separation, where that happens, is an estimate of your spatial resolution. Another way of saying it quantitatively is, if I can model it and it is absolutely a Gaussian, then I can give you the full width at half max of that Gaussian, and that's also an estimate of your spatial resolution of your system. So I put a point in What's the full width at half max in my picture? That's my spatial resolution. Okay. It doesn't have much to do with the pixel size. The pixel size is how you display your picture. I can display it with 10,000 by 10,000 pixels or 100 by 100. What is the underlying resolution is what is the full width at half max of a signal from this dot, right? So I need to use enough pixels to resolve that thing, right? And then you can just give that number and say that's what our spatial resolution is. For the GE scanner that we have in the Jacobs Hospital that does all of the cardiac stuff, we've measured it very, very precisely, and the full width at half max is about 1.2 millimeters in both X and Y, right? And you can put as many pixels as you want over that, but that's the underlying resolution of the scanner. Okay. Well, we've made up some time. Quality. Okay. We did these slides. We did these? We did not? Oh, okay. Thanks. Yeah. Yes. Yeah, that's one of the determining factors in CT. As you can imagine, if I have my detector along here and I have 10 elements, I'm not going to be able to resolve things terribly well, spatially. If I have 1,000 elements and they're all 1 millimeter, then when I look and project down, I can identify where something came from with higher fidelity. We'll get into like how that works. It also depends how many projections you take, right? It's like that. But principally on CT, it's like how fine is your sampling grid, right? Okay. Well, if you guys haven't seen this slide, then we're going to look at it. So just the way we looked at the phantom itself, this is a sort of cartoon of what's going on. So I have an input signal at a specific spatial frequency. So this is 1 cycle per millimeter. So here's a millimeter. I have 1 cycle of brightness in that millimeter. That gives you a spatial resolution, right? And when I pass it through my imaging system, the picture I get has the same frequency. Thankfully, my imaging system doesn't shift the frequency, which would be very complicated, right? It's the same frequency comes out, but the amplitude is reduced a little bit. And then if I double that frequency, right, same frequency comes out, but now I'm down at 56% of what I started, and it goes from this pattern to this pattern, right? So here's what went in, this thing. Well, this is a square wave, this is a sine wave, sorry about that. This comes out. And so basically the distance between the trough and the peak has been reduced, right? Just in terms of brightness. So it's been reduced here. Higher frequency, now I'm at 4 times where I started. That frequency comes out with very low contrast. So we're down to 13% of the maximum contrast until finally, if I go to a higher frequency, I just get an average value across there. I don't even resolve it anymore. So now I'm beyond my ability to resolve something. So this is called the modulation transfer function. When we look at a specific spatial frequency that is going into my system with one of these patterns, and I plot the reduction in amplitude at that frequency. So here, this is spatial frequency along here, 1 cycle per millimeter, 2, 4 cycles per millimeter. And then this is the relative brightness or the relative trough to peak difference, contrast for that pattern, okay? Just, it's this from here to here, right? So they're dropping as we go. So over here, I'm down at 13%. You can do this for every spatial frequency and map this function absolutely accurately. And that's called the modulation transfer function. Okay, so how do I go from I transmit all of the information to when I can't transmit any of that information because the pattern's has too much resolution. Yeah. Each modulation transfer function is basically measured for every imaging machine you have, right? So not only the imaging machine, the protocol you're using on that machine. So you can change the reconstruction kernels and stuff, and the modulation transfer function will change, right? So some modulation transfer functions, if you reconstruct one way, will provide much better, higher spatial resolution and roll off farther out here. It gives you higher spatial resolution pictures, but it also gives you noisier pictures, as we'll see, right? So this function itself is what you use to completely characterize that imaging device and that protocol for using the imaging device.
So it's like a tool to say this is a verifiable method of getting an image for a certain goal.
Absolutely. And what this also is used to do is to report, if I build a system and I come up with a reconstruction technique and then I want to sell it, I can tell people, they ask, well, what's the spatial resolution of the system? And you can show them this graph, right? And then that's fundamentally what that resolution is. And if you have two systems, you can measure it on one, measure it on the other, and then put the two graphs on top of each other as like a competition, right? And say which one gets me out farther in spatial resolution. What is the shape? Does it go down really quickly? Does it extend out far? So this gives you the whole deal, this modulation transfer function. Sometimes the manufacturer, what they will give you is, say they'll pick 10% and they'll say, we cross 10% at 4.38 cycles per millimeter. And that's our spatial resolution. And some of them will say, we cross 1% at blah, 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 right? So it tells you where the thing goes out to. Yeah. Oh, the 1 is a relative number. So basically, if I image something that has very low frequency, so it's a sine wave that I can absolutely resolve, then that's my starting amplitude. And then everything else is divided by that to get the drop off, right? So it's just relative. Right? Yeah.
Yeah, are always that shape, and like what are some things that would determine for this curve, like what a image is?
Yeah. So the question is, does the MTF always look like this? And what makes it look different? And the MTF usually looks like this in the sense that it's a blob that decays away, right, as you get to higher frequency. Normally, that's what they look like. It would be very rare, although it happens, that you would start here and it would get high, it would come up at a specific frequency and then roll down. But you can imagine that could happen. You suppress lower frequencies and amplify higher frequencies. It can go farther out depending on your reconstruction technique, and then roll off more aggressively down here. So it has all different types of shapes depending on how you reconstruct the image from the raw data. Now, the fascinating thing about the MTF also is that as you can see, this function looks kind of like a Gaussian, right? It has a full width at half max and kind of like a Gaussian. If I take the Fourier transform of this thing, what do you think I get? You get another Gaussian, exactly. And that other Gaussian is the point spread function. So basically, if I look at my point spread function and it's a Gaussian, if it's really, really narrow in space, like the full width at half max is like 100 microns, so I have a really narrow point spread function, then when I look at the modulation transfer function, It's very broad. It's very wide. As that point spread function gets wider and wider, I get blurrier and blurrier pictures. The spatial frequencies roll off more quickly. So this thing gets narrower. So the point spread function and the modulation transfer function are Fourier pairs, and they behave just as Fourier pairs do, right? As one gets wide, the other one gets narrow. Okay. But we'll look at that. You can look at that mathematically. Okay, we did this. Here's the setup. I didn't show this picture before of this thing here is a tissue equivalent material that you put in the scanner. If you want to image a phantom in there, if anybody has a project and you think up, I'd like to do a phantom image, and these are the images I would need. We can get those for you if you want to do that. Okay, so we just go in, and the thing about CT is that you go in and you set things up, and you're ready to do the data acquisition. You go, boop. Well, I guess we're done here. It's like a second. Whereas MR, we'd spend the whole damn weekend in there. It just takes forever to do anything. So CT is great that way. So yeah, you can make different pictures with different acquisitions, different imaging kernels, different materials in there, stuff like that. So this thing here is tissue equivalent material such that you get a realistic number of photons at your detector. If you image something like a phantom in air, it looks spectacular because you get a lot of photons, right, on your detector. If, on the other hand, you put it in something that is like a 160-pound person, then the number of photons goes way down. So your realistic image quality should be done with these things always. So this phantom is inside here, right? And then we went through that. Okay. Hey, look, this thing here is one of those little beads. It's a 0.38 millimeter pen tip. And it's on this is the phantom here, and this is the insert. There's a little gap so that you can put it in. And these beads are just like Scotch taped on the outside of it. So we'll zoom that up so you can see what the bead looks like. It's tungsten, I guess. So it's like it absorbs a ton of X-rays, right? So it's very, very bright, right? A lot of X-rays get absorbed by that. This is all CT, yeah. Yeah. So basically, the first half of the class, and unless I say this is MR, it'll be CT usually. Okay.
So given something like MR, is there kind of like a trade-off in like time of image acquisition and then your spatial resolution?
Yes. So the question is, in MR, and we can say CT as well, is there a trade-off between the time you're willing to image and the spatial resolution you will achieve? In MR, that is absolutely true, okay? Because as we acquire data in MR, usually the way you acquire the data is the blurrier stuff first, and then as time goes on, you collect data that's of higher and higher spatial frequency. The interesting thing about MR is that theoretically, you can keep cranking your gradients and getting higher and higher spatial frequency out to like tens of microns, right? However, to get out there and get signal out there takes a long time, right? And you have to average a lot of signal together. Eventually, what happens in MR is if you claim I'm making an MR picture and it has 10 micron by 10 micron voxels, 10 by 10 by 10, depending on how long it takes you to image the signal from that voxel is quite important because the protons themselves will diffuse out of the damn voxel, right? When it's at 10 microns, right? If you think about, well, how big is a cell? How does water diffuse around? And so eventually, the signal that you're imaging as you crank your gradients up and up and up, it just disappears because all the protons diffuse out of the thing you're trying to get them. So, but that's advanced MR. However, diffusion was first measured in test tubes with NMR. So the people doing spectroscopy are saying, well, how does diffusion affect our signal? And so they figured out a way. These two guys, Steskel and Tanner are their names, wrote this great paper about, if we turn on, and these are the shim coils again, if we turn on the gradient really strong, our signal diminishes because of this diffusion effect. The random motion of the protons in this gradient causes our signal to go down. And it's a beautiful paper. I'll post it up. And in that paper is an equation. This is a paper from the 60s. In that paper, there is an equation which is the MRI imaging equation. It is how MRI imaging is done. It's just sitting right there. And after learning MRI imaging and going through all that data, and then you go back to this paper, go, oh my God, it's right there. Right? So it's pretty interesting. They didn't stop. They were interested in other stuff. Hello again. Okay, so we're going to look at contrast now, which is like, what is the difference in brightness between two types of tissue, and can I determine that they are different, right? So here, this is out of a paper. There's a connection to the paper here. This is a CT image of a liver, okay? And this person has a primary cancer at a different part of their body. And the question is, do they have a metastasis in their liver, right? And it turns out that on a basic non-contrast CT scan, the metastasis will show up as slightly darker than normal liver background because it's a slightly denser material, right? So everyone looking at that, I don't know whether or not, let's get here. Hey, can anybody, so you all know where the liver is, right? On the top left. It's a really large organ there, right? It's got some vascular structure going through it, right? Anybody see the Met? Yep, exactly. So that thing right there, that little dark spot, is the metastasis. So is spatial resolution our issue here? No, right? We have enough spatial resolution to resolve that darn thing. What we need now is contrast, right? I would burn spatial resolution to get more contrast such that I could detect this thing, right? And so there's a couple of ways to define contrast in CT. One is a local contrast where you look at the fractional change between your target, which in this case would be basically our MET, which is down here, and the background, which would be the normal liver. So you look at the difference between those two divided by the background, right? And that fractional definition of contrast is particular to CT because of the way CT physics runs. It simplifies the math of what the contrast is. However, when the rubber hits the road and we actually look at measuring whether or not you can see this thing, I usually use just contrast to noise definition, which is what is the brightness of this minus the brightness of that divided by the noise, right? Just raw signal. And that'll be in your problem set. So here you can see in this image of that phantom that we've been looking at, let me see if I have, no, here you definitely can detect that there's a hole there, and it's not a spatial resolution thing, it's a contrast thing, right? Noise, going from left to right here, this is what a scan looks like when you just increase the noise. And in this case, what we're doing is we are adding random numbers to this picture, and we're increasing the amplitude of the difference between the max and the min of those random numbers as we go from left to right, okay? And true noise, if it's white noise and it's ergodic, means that a pixel here which is just background noise, has absolutely no correlation to the pixel next to it, which has no correlation to the pixel next to it, right? So that's just random noise. That's sort of in the background noise. And then as the ratio of the amplitude of that background noise gets higher, or as its amplitude comes up, we see that the ratio of the signal to that noise is decreasing, and finally it's messing up our actual signal. So sources of noise in MRI, you have noise of your electronics. Principally, most of the noise in MRI comes from the patient. So it actually turns out if I have a receiver coil and the patient's in the scanner and I have it in my hand, I look at my scope, right? Obviously, I've got 0 voltage, nothing happening in this receiver coil, this antenna. But then when I crank the amplitude up and up and up and up, finally, I start seeing it's just a random noise signal, right, in time. And that's the thermal noise of the resistors and everything in that electronic device, right? If I dropped it into liquid nitrogen, that amplitude of noise would go down because its temperature would drop. But at room temperature, it has a certain noise level. In MRI, when I take that antenna and I want to image the patient, and now I put it right on the patient's chest, the noise goes up because it's measuring random thermal motions of charged particles in the patient, right? So you get this noise signal from the patient, and that is principally the biggest source of noise in MRI, is the patient's own thermal motion, right? In CT, the noise comes from background noise from electronics, but with photon counting CT, that kind of goes away. We'll get there. But also, the principal source of noise is the uncertainty in the number of photons you have detected in each detector. And so, as you can imagine with CT, you want to use the lowest amount of photons possible to minimize the radiation that is the patient sees in making that picture. To do that, on the detector, there are places in which you are down to like 5 photons, in a detector, right? So you just keep reducing your dose until you get down to there, right? And so you're always looking at uncertainties of like, do I have 5 photons? Do I have 6 photons, right? Now, there are other places where you have tens of thousands of photons, right? In like air and stuff like that. But in really thick pathways that are going through a lot of tissue, you can get down to low numbers. It turns out, as we'll see, you take the logarithm of that number of photons divided by the number of photons that you would have without a patient in there. And when you take that log, obviously, as we get down towards 0, you know how the log goes, right? As you get to 0, it just takes off to infinity. So as we get down to those lower numbers, the uncertainty in the amplitude for our reconstruction gets really high, right? So that is why they always futz around with the raw data to make sure they don't have, detector elements that only have a few counts in them. And so here's the basically increasing noise, increasing blur. So we can get a higher signal to noise picture by increasing the blur. We just average pixels together, for example, so that when I measure my signal amplitude over the noise, I can get a higher signal to noise picture. But, when I look at the detail of this, the same amount of blurring comes here. You lose high frequency information in doing that. Usually, though, it's a trade-off. If you show a radiologist all of these pictures and say, which one will you accept? Like to do your diagnosis, which one of these will you accept? I'm sorry, this one is unavailable because it costs too much, right? For whatever reason. So where do we go along here, right? And so they can say, oh, I'll be okay with a bit more blurring because I don't want to go over here where it's noisy, right? So that's where it's an interactive process that you work with someone reading the pictures to determine where they want to operate, you know. It's both. Reconstruction can smooth the picture a lot and get rid of noise. So you can crank that reconstruction smoothing up. And also, by the way you take the picture for CT, I just use more radiation. That improves things. MR, I use more time. Right.
So, but given certain constraints on like patient and machine, you kind of have to maybe sacrifice some of the way that you take the image.
Yep.
Maybe do more work on a little bit better at.
Well, yeah, you optimize all that stuff and you do it with the radiologist in the loop. If you don't do it with the radiologist in the loop or the cardiologist, they will tell you, right? You send them a picture, it doesn't take long. The phone rings, they say, what the heck is this? Nope. Yeah, you got to do that again, right? So, all right, great.